The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Dr. Valerie W is a professor of biochemistry and molecular medicine at the George Washington University School of Medicine and Health Sciences in Washington, D.C., as well as the mother of a son with autism spectrum disorder. In late 2004, because of her personal interest in autism, she redirected her research focus and has since become a leader in the application of multidisciplinary integrative genomic approaches to ASD, which involve the integration of large-scale data from gene expression, behavioral, genetic, and epigenetic analyses. Currently, she has turned her attention to environmental contributors that may increase risk for autism through alterations of the epigenome that may be responsible, at least in part, for the heritability of autistic traits. These webinars are made possible through generous donor support. If you'd like to contribute, please visit our website at autism.org.
Thank you, Denise, for inviting me to present my research at this webinar. What I'm going to do today is kind of describe a path that we've taken that started out with doing some integrative genomic studies to try to understand the biological basis of autism to where we are today in looking at a certain class of environmental contributors to risk for autism. So in today's talk, what I'll do is describe how our integrative genomic studies on autism spectrum disorder has led to our investigation of endocrine-disrupting compounds, or EDCs, as potential environmental risk factors for autism. Then I'll present preliminary findings on the impact of a specific EDC, the herbicide Atrazine, on gene expression in a neuronal cell model. I'll describe our current studies on epigenetic changes, that is DNA methylation, in human sperm in association with environmental exposures to long-lived endocrine disruptors. And then we'll discuss how epigenomic alterations caused by EDC exposures may contribute to elevated risk for autism. So first of all, what is autism? I'm sure most of you know this, but just to get everyone on the same page, autism spectrum disorder, or ASD, refers to neurodevelopmental disorders that are characterized by core deficits in two main symptoms. First of all, reciprocal social interactions and social communication, as well as restricted repetitive stereotype behaviors. It used to be that language development and usage was part of the triad of symptoms that marked autism spectrum disorder, but now that has been sort of wrapped into social communication. And it used to be that autism used to be divided into three classes: classic autism for the most severe, PDD, NOS, that's pervasive developmental disorder not otherwise specified to describe a moderate phenotype, and then Asperger's syndrome to describe the so-called high-functioning form of autism. All of these different terms now are housed within the umbrella term of autism spectrum disorder, or ASD. But regardless of what you call it, autism is a significant public health problem worldwide. The prevalence, according to the recent CDC estimates, is 1 in 59 individuals with a male to female ratio of about 4:1. The estimated lifetime costs for raising a child with autism estimated in 2017 is $2.4 million per individual. This is in the US. So our interest is in what causes autism, and to help my lab kind of organize our thoughts as to the many different factors that go into understanding of the etiology of autism, I assembled all of these components into what I call an autism pyramid. At the very base of the pyramid, I've placed the ASD phenotypes. These are the observables, including behaviors, symptoms, as well as brain circuitry, which can be observed by specialized neuroimaging techniques. And then each layer above indicates something that controls everything, all of the layers below it. So that immediately above the ASD phenotypes, I have gene expression profiles, which is the level of gene activity, which determines the actual functions of the cells in the tissues that ultimately give rise to the different behaviors and symptoms. Above that layer of gene expression profile, we see genetics and epigenetics side by side because they're equally important. I think, as many of you know, genetics is encoded in our DNA, and that can be considered the hardware or the directions for the genes, all the genes that are encoded in our cells. Whereas epigenetics refers to the regulatory mechanisms that determine when and where the genes turn on or turn off. And you can consider that the software. The software is equally important to the hardware in that it determines the regulation of those genes. And there are different forms of epigenetic mechanisms, including DNA methylation, which I'll talk about later, microRNA expression, histone modification, and chromatin remodeling. And at the very top of the pyramid, I've put all of the environmental factors that we really don't understand much about. And these triggers can be intrinsic, that is internal for the organism, such as hormones or inflammation, which is part of our bodies, or it can be extrinsic environmental factors, such as pesticides that we would find in our environment. Okay, so our interests in autism are the following. The goals in my lab are to identify genes, biological pathways, and functions that can be used for targeted therapies, to identify diagnostic biomarkers of ASD, and to develop a systems-level understanding of the pathobiology of ASD. One of the main problems with studying autism is the heterogeneity in the manifestations of autism. And this I liken to asking a scientist to study a bowl of mixed fruit. It really depends on what fruit you happen to pick out of the bowl in terms of this description. So we try to solve that problem by reducing the clinical heterogeneity in autism using scores on a commonly used diagnostic test to subgroup individuals on the spectrum according to the severity of behavioral symptoms. Our underlying hypothesis for all of the work in my laboratory is that autism is a systems disorder with severe neurological manifestations, and therefore can be studied using more easily accessible tissues such as blood, which may reflect the molecular and functional abnormalities in the brain, as well as reveal biomarkers for diagnosis. As I mentioned earlier, our experimental strategy is to divide and conquer. That is to reduce the heterogeneity in autism by using scores on a commonly used diagnostic test called the ADIR, Autism Diagnostic Interview Revised, which is used to subgroup individuals according to the severity of the behavioral or clinical symptoms. The ADIR is considered the gold standard behavioral diagnostic for autism. It is a detailed questionnaire comprised of over 100 different questions that probes the subject's functioning across multiple behaviors. This questionnaire is usually administered to parents by a neuropsychologist. And the questions probe behaviors such as the child's spoken language abilities. This includes ability to chat, use of echolalia, pronomial reversal, the child's nonverbal communications, that is the use of gestures, head nods, etc., the child's social functioning at a higher level, such as interactive play or the ability to offer comfort to others, repetitive and stereotype behaviors. These are the most observable behaviors, such as rocking, flapping, or lining things up. The child's restricted interests, perseverance on a topic or in certain objects or parts of objects. And then, interestingly, savant skills, such as visual, spatial skills, memory, reading, music, art, and math. And savant skills are kind of an interesting phenomenon that is present in individuals in autism at a higher frequency than in the general population. So taking 123 item scores from the ADIR test, diagnostic test, we were able to separate individuals on the autism spectrum into 4 clinically distinct phenotypes by means of cluster analyses of the raw scores. What you see here in this figure are 4 groups of individuals represented by the 4 plaid blocks that you see here on the screen. And each horizontal line on the block refers to one individual, and each vertical column refers to one of 123 ADIR questions on this behavioral diagnostic. And the color red or black refers to the severity scores, where bright red equals a severity score of 3, which is the most severe score on the particular ADIR item, and a score of 0 is represented by black color. Now, what you can see by eye, I think, looking at the 4 blocks, is that they are just intuitively different, just in terms of the patterns. And that in the first block that I named severe language impaired, there's a broad red band in those columns. And all of those columns refer to spoken language items on the test. Whereas if you look at the columns on the extreme right of each block, those items represent the savant skills items. So according to the different columns that are indicated on these 4 blocks, I consider the first block, one of the blocks with the broad red band, the severely language impaired group, the block below it with higher frequency of savant skill items checked in red, the savant group, and then the other 2 blocks on the right, the intermediate group, that is intermediate in severity between the mild, the group below it, which has more black, indicating normal functioning on a number of items there. And if by doing this, we're able to do another type of cluster analysis called principal components analysis that show that without any kind of direction by the experimenter, which is myself, these different individuals, these are almost 2,000 individuals, can be clustered into different subgroups according to their clinical phenotype, where you see the red dots indicative of people that are represented in the first block under severe language impaired, and so forth. So bottom line is, by doing cluster analyses, we are able to divide the autism population into 4 reasonably distinct clinical phenotypes of autism. And the reason for doing this is that we're interested in using large-scale genomic methods to try to understand the biology of these different subtypes of autism. And the first studies that we did on the genomic scale was that of gene expression profiling, which is looking at the gene activity levels in groups of individuals with autism in the different subtypes that I've just showed you in comparison to non-autistic controls. And so the questions that can be asked by such a study is, what genes are differentially expressed between autistic individuals and controls? Can the different subtypes of autism, as we've defined them, be distinguished by different gene expression profiles? What do differences in gene expression profiles tell us about the respective biology of the different subtypes of autism? And finally, can these differences be used to discriminate between autistic individuals and controls? That is, can they be used for diagnostics? So first of all, what I'm showing you here is the results of gene expression study, in which, if you're looking at the upper figure here with the red and green blocks, we can tell that according to gene expression levels, where each horizontal row now is a gene and each column is an individual that's divided into these different subgroups of non-autistic controls, the severely language impaired group under the red bar, the mild individuals under the blue bar, and then those with savant skills under the yellow bar, we could see that there are different patterns of red and green amongst the genes, which indicates the green being lower levels of expression relative to a universal standard. Red indicates higher levels of gene expression according to the same universal standard. And there's the biggest difference in gene expression between the severely language impaired group and the autistic controls. But as you go down towards the milder end of the spectrum, you can see that the color of the gene expression tends to revert back towards the controls. That is, towards the milder end, you begin to see expression patterns that approach that of the controls, even though those individuals under the mild and savant categories are also autistic. If we do a different kind of analysis called a principal components analysis based upon this gene expression profiles, we can see that the different subtypes of autism can also be separated into these subgroups of yellow dots, red dots, dark blue dots, in comparison to the turquoise controls there. So there are distinct groups that can be separable according to gene expression profiles. And so the next question was, what do these different gene expression profiles tell us about the biology of the different subtypes of autism? Here, we're looking at a comparison between the genes that are differentially expressed between the severe language impaired group and controls. So there are over 4,000 genes that are differentially expressed. And then for the mild subgroup, there are about 500 genes that are differentially expressed between the mild subgroup and the controls. And for the savant, 127 genes that differentiate the savants from control group. If we look at the functions of the different genes that, for example, are overlapping between the severely language impaired group and the mild group, and look at those functions, we see that those 199 genes are associated with cell death, with inflammation, embryonic development, synaptic transmission, neuronal migration, memory learning, muscle tone, as well as head size. And all of those associations do relate to what we know about autism. On the other hand, if we look at the genes that are unique amongst the language impaired subgroups, such as the 3,900 genes, and look at those functions, we see an overwhelming number of cell death genes, as well as genes that associate with a painful response to normal sensory stimuli, muscle rigidity, to heightened sensitivity to painful stimuli. So in this group of genes, there are genes that associate with abnormal sensory perception, also genes that associate with epilepsy, and circadian rhythm genes, which are not seen in any of the other two subtypes, the mild or the savants. And if we look at the 20 genes at the center of the Venn diagram, we see, surprisingly for us and puzzlingly, we found 20 long non-coding transcripts, of which many are responsive to regulation by male hormones. We do not know at this time what those genes do, but it's intriguing that with the male bias in autism, that at least 6 of those are regulated by sex hormones. Among the genes that are differentially expressed, the circadian rhythm genes really piqued my interest because I had not associated circadian rhythm with autism in the past. So looking more closely at those 15 circadian rhythm or clock genes in the severe autistic group with the language impairment, we constructed a network to see what are the functions associated with those specific genes. And we see here by this rather complicated network that in addition to circadian rhythm genes, as well as sleep, which of course is related to circadian rhythm, we see other functions that are associated with this set of 15 genes, including digestion, which is implicated in individuals with GI disorder. We see neuron toxicity, muscle function. A number of people with autism spectrum tend to have hypotonia, also in genes associated with inflammation. And one of the genes that I've circled in the middle is called RORA, R-O-R-A, and I circled that because this has kind of helped us link autism to the environment via the effects on this particular gene called RORA, and I'll get to this later. So to summarize the functional analysis of the circadian rhythm genes, we find that the associated functions and disorders include the sleep-wake cycle, not surprisingly, memory, learning, cell proliferation, steroid biosynthesis, as well as digestive disorders, inflammation, muscle dysfunction, and neuron toxicity. what we were hoping is that some of these dysregulated genes might point us towards novel target genes for subtype-specific treatment. One of the genes, called AANAT, is a rate-limiting enzyme in the biosynthesis of melatonin, which is a sleep-inducing hormone. So a deficiency, as we see here for this particular subgroup of individuals, suggests that maybe this group of individuals, particularly if they're plagued by sleep disorders, might be helped by melatonin supplements. Another gene in this particular subset is the gene for DPYD, which is an enzyme that is necessary for the breakdown of pyrimidines, which is part of DNA, a component of DNA. Its deficiency leads to reduced beta-alanine, which is a known neurotransmitter. And a genetic deficiency, a genetic mutation in this particular gene predisposes to epilepsy, mental and motor retardation. And in fact, 18% of people with a genetic mutation in DPYD have ASD. So a deficiency in this particular gene might suggest that perhaps beta-alanine supplements might help this group of individuals, or that maybe because of the association with epilepsy, maybe anticonvulsant medications might be prescribed as the first line of therapy or targeted therapy for this group of individuals who exhibit a deficiency in this enzyme. So to summarize then, this type of approach is what we hope to learn from these type of genomic studies on the biology of autism, and would lead us to what we hope for is the field of personalized medicine, that is treating individuals according to their specific deficits with respect to autism. So this general approach of dividing the autistic subgroup into these different subtypes has also helped with respect to genetic analyses, which I'm not going to describe here. But when we did do this type of subtyping, we could identify 18 novel SNPs, genetic variants, that were missed if we do not do the subtyping. But right at the moment, I want to switch gears and talk about our epigenetic studies, that is specifically DNA methylation. We're asking the question, are epigenetic mechanisms, do they contribute to gene dysregulation in autism? And here I just want to remind folks that epigenetics does not involve a change in the DNA sequence, but rather, in the case of DNA methylation, it involves putting a chemical mark on a particular base in DNA that can alter the function of that particular gene by either turning it on or turning it off. So to determine whether or not there were epigenetic changes that could be associated with autism, we used monozygotic twins who were discordant for the diagnosis of autism. And here we're looking both, for this set of twins, both at gene expression differences, which I've mentioned earlier, as well as methylation differences. So in this study, we involving discordant twins as well as SIB pairs, we looked for differentially methylated genes amongst this discordant twins and SIB pairs, as well as for differentially expressed genes. We looked for the genes that overlap between these two groups, that is, the differentially methylated genes and differentially expressed genes, for genes that could for differentially expressed genes that could be regulated by methylation. We do pathway analysis to identify functionally significant candidate genes, and then select a couple for confirmation of methylation and expression in the lymphoblastoid cell lines, which we are using as a cell model for our studies. And then we take it to the brain to evaluate changes in the protein level of the select genes in the brain tissues of individuals with autism versus non-autistic controls. So to summarize our methylation data, we found a set of about 25 genes whose expression levels could be related to higher methylation of those genes. And if we do a pathway analysis, we identified a number of genes that are regulated by methylation that are involved in steroid biosynthesis, apoptosis, inflammation, digestion, mental deficiency, fetal development, etc. We then go on to confirm a couple of these genes, BCL2 and a gene called RORA, or RORA, for differential methylation and expression in the lymphoblastoid cell lines, and then look to the brain for changes in the expression of both of these genes. Now, I will focus here on RORA because this was a gene that has never before been related or associated with autism. We found this to be an interesting gene for the following reasons. First of all, at the time of our studies, we learned from the mouse models of RORA deficiency that this gene, RORA, protects the brain against oxidative stress and inflammation. This gene also regulates circadian rhythm, and the loss of RORA leads to developmental defects in the cerebellum and loss of Purkinje cells. Furthermore, RORA-deficient mice are sexually dimorphic with respect to production of cerebellar neurosteroid and Purkinje cell survival during aging, with male mice being more severely affected. Finally, RORA deficiency results in ataxia and hypotonia, that is low muscle function, as well as perseverative behaviors and deficits in spatial and discrimination learning. The implications for autism of this set of properties of this gene is that neural inflammation and oxidative stress have been detected in the postmortem autistic brain. So this links to RORA being protective against oxidative stress and inflammation. Furthermore, circadian rhythm genes are implicated in autism, not just by our studies, but also by some genetic studies as well. Purkinje cell deficiency has been the earliest noticed and most consistent neuroanatomical abnormality in ASD, and RORA is important for Purkinje cell survival in the cerebellum. And finally, males are more affected by ASD than females, thus referring back to the sexual dimorphism of RORA in the mouse. Okay, so the questions we had regarding RORA's involvement in ASD are, what are the regulators of this gene RORA, and what does RORA regulate? Given the sexually dimorphic characteristics of RORA-deficient mice, we wondered whether or not RORA might be differentially regulated by male and female sex hormones. And then we also wondered whether RORA deficiency might be evident in brain tissues from individuals with autism. And since RORA is a nuclear hormone receptor which acts as a transcriptional regulator, that is, it can regulate other genes, which genes are transcriptional targets of RORA, and how do they relate to autism? So to address these questions, we first looked at the regulation of RORA expression by male and female hormones. And as you can see in this slide, estrogen increases the levels of RORA expression both in a concentration-dependent and time-dependent manner here, whereas testosterone or dihydrotestosterone, a derivative of testosterone, tends to decrease the level of RORA expression both in a concentration-dependent and time-dependent manner. Then we wondered whether or not RORA expression might be reduced in the autistic brain. And at the same time, around the time we were doing these studies, we found another study on cancer saying that another gene called aromatase is a regulatory target of RORA. So taking a human postmortem brain from autistic individuals and controls, we looked for the levels of RORA as well as aromatase protein in the brain using immunofluorescence microscopy. And what we found is that there's a statistically significant decrease in the levels of both RORA and aromatase in the brain tissues of autistic individuals. And in fact, aromatase is strongly correlated with RORA, almost in a 1-to-1 ratio, with the R-square being 0.91. Okay, you might say, well, so what? What is the relevance of decrease in aromatase? Well, the autistic male to female ratio is at least 4-to-1. And Simon Baron Cohen at Cambridge has been a strong proponent of what he calls the extreme male brain theory of autism. In a nutshell, what this theory suggests is that elevated fetal testosterone poses increased risk for autism. However, there is a big black box connecting elevated fetal testosterone to risk for autism. So there was no molecular explanation for the higher testosterone levels or for the significant male bias in autism. Aromatase is an enzyme that converts testosterone to estrogen, estradiol. And so from our studies, we could see that if RORA and its target gene aromatase are knocked down in autism, the decreased aromatase would give rise to higher levels of testosterone, thus offering a plausible explanation for the elevated fetal testosterone. So we've developed a model to describe the sort of a feedback mechanism by which DNA methylation acting upon RORA could decrease RORA levels of RORA in the brain, leading to decreased levels of aromatase, increasing the levels of testosterone and its metabolite DHT. The increase in DHT would further decrease the level of RORA expression, being an exacerbating factor. On the other hand, we propose that females with higher levels of estrogen might be somehow protected against agents that might decrease RORA, because estradiol or estrogen will tend to increase the expression of RORA. And what we found by looking at RORA expression in the frontal cortex of males, both mice and men, we find that there are lower levels of RORA expression in the frontal cortex in males. And this is consistent with this model. So then, as I said earlier, RORA is a nuclear hormone receptor, which means that it can regulate a number of other genes. But the question was, what are the genes that RORA regulates? And what we found by doing a chip-on-chip analysis to look for the genome-wide targets of RORA, we found that there were over 2,500 potential target genes of the RORA gene. And over 400 of these are already known autism risk genes. And these autism risk genes function in neurogenesis, synaptic transmission and plasticity, axonogenesis, cognition, learning, and memory. We did additional analyses to further confirm a half dozen of these RORA targets that are shown below, such as RBFOX1, aromatase, which I mentioned earlier, another enzyme that also converts testosterone to estradiol, another gene called ITPR1, which acts at the synapse, neuroligin 1, an adhesion molecule also acting between nerve cells, and NTRK2. All of these were validated as RORA targets, and all of these are known autism risk genes. The functions of these genes are shown below, and they involve synaptic transmission, neurogenesis, mitochondrial integrity, synaptic plasticity, adhesion, axon guidance, etc. So bottom line is that if we find that any mechanism that disrupts RORA expression, including environmental factors, could increase risk for autism. And this leads us to our studies on environmental factors that might elevate risk for autism. So the impact of sex hormones on RORA expression suggests that RORA may also be dysregulated by endocrine-disrupting chemicals. Endocrine-disrupting chemicals are compounds that either mimic endogenous hormones or antagonize their actions, metabolism, or transport, thus interfering with normal hormonal activity and homeostasis. So we wondered whether or not RORA might be a target for gene-environment interactions involving EDCs that could increase risk for autism. In other words, can RORA expression be a canary in a coal mine to tell us whether or not a particular environmental chemical might pose risk for autism through dysregulation of RORA expression? So what are the EDCs? Here are a few examples of endocrine disruptors. Among them are Atrazine, which is in a lot of different herbicides, bisphenol A, which is found in plastics, dental sealants, paper receipts, something that a lot of you have heard of, is BPA. DDT, which is a long-lived breakdown product, and its long-lived breakdown product, DDE, which is found in pesticides. Although these are now banned, these pesticides have a very long half-life in the environment. Other EDCs are phthalates that are found in soft toys, flooring material, even cosmetics. Polychlorinated biphenyls, or PCBs, which have been used in coolants and lubricants. Polybrominated diphenyl ethers, used as flame retardants in textiles. Valproic acid. Certain drugs are also endocrine disruptors. Valproic acid is a drug used for epilepsy, bipolar disorder, and major depression. The major concerns regarding EDC exposures are the effects of cumulative exposures from persistent organic pollutants, for example, DDE, PCBs, and BDEs, as well as epigenetic changes, particularly when they occur in germline cells, that is sperm and egg cells, where these changes could be propagated transgenerationally, that is from generation to generation, through the DNA in eggs or sperm. Okay, so to briefly describe our studies on atrazine, first of all, atrazine, as I said, is found in many herbicides. The EPA limits for a 90-day average of atrazine is 37.5 parts per billion, which translates to 175 nanomolar. It's currently accepted as safe in community water systems. This chemical is easily absorbed by the GI, the gastrointestinal tract, the lungs, or skin. And literature reports the effects of atrazine on sexual differentiation in wildlife, with a lot of mutations observable. But with respect to human effects, there's a study published in 2009 that shows a correlation between the levels of atrazine that's measured in surface water around the country by month of the year and the number of congenital birth defect rates by month of conception. And you can see that there is kind of a parallel course of birth defects that track the levels of atrazine in the surface waters. And although this does not prove that atrazine is causing the birth defects, this is a very interesting correlation to see between the birth defect rates and the levels of atrazine available in the surface waters. So with this in mind, we decided to look at the effect of atrazine on a gene expression of RORA in a neuronal cell culture upon short-term treatment. So here we're treating neuronal cells in culture with different amounts of atrazine for only 2 hours, and then we're looking at the levels of RORA expression. And what we found that at as little as 0.1 nanomolar atrazine, we see a significant increase in the level of RORA expression, whereas at 10 nanomoles atrazine, we see a significant decrease in the level of RORA expression. And if I go back a slide, we can see here that 5.5 nanomolar is the level of which there is a maximum amount of atrazine in the surface water, and that's comparable to this levels of RORA here between 1 and 10 nanomoles in our study. Then we did gene expression profiling using Affymetrix human transcriptome arrays at 2 concentrations of atrazine, at 0.1 nanomolar and 10 nanomolar atrazine. And we looked at the different numbers of genes that are differentially expressed by in cells that are treated with atrazine at 0.1 nanomolar or 10 nanomolar atrazine. And we overlap that with the RORA targets that we have identified by our chip-on-chip studies. And if we look at the intersect between those genes that are differentially expressed between the 2 different concentrations of atrazine here in the middle, we see that the pathways affected that is implicated by those genes are axon guidance, glutamate receptor signaling, chemokine, notch, and ephrin receptor signaling pathways. And the neuronal functions that are affected, that's implicated by those genes, are development, growth, morphology and guidance of neurons, neuritogenesis, synaptic development, and transmission. If we look specifically at the 22 genes that are also RORA target genes, we see that the associated functions are migration and cell death of granule cells in the cerebellum, formation of the brain, seizure disorders, and mental retardation and movement disorders. So the take-home message from this exploratory study on Atrazine is that dysregulation of RORA expression by endocrine disruptors, such as Atrazine, is a potential mechanism for gene-environment interactions that may increase risk for ASD by inducing a domino effect, leading to the dysregulation of transcriptional targets of RORA, as well as many other genes that may contribute to the neuropathology of ASD. Now, while our studies have focused on the immediate effects of EDCs, that is only after 2 hours, what are the long-term effects of EDC exposures, especially in vivo, and how might the impact of environmental agents be transmitted across generations? And for these questions, I turned to a study that was recently published in the literature by Michael Skinner's group. And this study is called Atrazine-Induced Epigenetic Transgenerational Inheritance of Disease, Lean Phenotype, and Sperm Epimutation Pathology Biomarkers. For this study, female rats were injected with atrazine during gestation for a very brief period, a 6-day period. And then male and female offspring from different litters of the exposed F0 females were bred through 3 generations without any further exposure to the chemical atrazine. And what was examined from this study, in this study, were various pathologies and phenotypes, including testis disease, mammary tumors in males and females, early onset of puberty, body weight, as well as DNA methylation in the sperm from the male offspring in 3 generations, the F1, F2, and F3 generations. So the results are that in F1, the immediate offspring of the treated females showed no disease but lower body weight. And then in the F2, F3, there was increased frequency of testis disease as well as other phenotypes. But most important or significant as far as I'm concerned is that there was DNA methylation in sperm that was increasingly altered in the F1 to F2 to F3 generations, with the number of genes increasing in each subsequent generation. And my analysis of this data, the methylation data, showed that there was significant enrichment in ASD genes, particularly in the F2 and F3 generations. So with that, we turn to looking at the impact of endocrine disruptors on the human sperm methylome. So the specific aims of our study were to investigate DNA methylation in association with exposure to high and low levels of the compound DDE, which is a long-lived and persistent breakdown product of the DDT pesticide. Then we wanted to determine the pathways and functions affected by differentially methylated genes, to determine whether or not the differentially methylated genes are enriched in autism risk genes. So for samples, we used semen from young adult men in the Faroe Islands, which are in the North Atlantic. These are islands owned by Denmark. And the people in these islands have a natural diet of pilot whale meat and blubber, which exposes them to high levels of endocrine disruptors, particularly the PPDDE, as well as other endocrine disruptors such as PCBs. The experimental approach was to use whole genome bisulfite sequencing of sperm DNA, and this is shown in the schematic below. So we take sperm, isolate DNA, perform bisulfite conversion on the DNA, and then subject the DNA to whole genome bisulfite sequencing. From the data of the WGBS, percent methylation was calculated at each of the cytosine sites in the DNA, and differentially methylated regions were identified. Differentially methylated regions were then analyzed for genes that correlated with autism risk genes. So here we see a heat map of the differentially methylated regions from our discovery set of 32 samples. And what we can see here from these two blocks, that are shown, you can see the color differences here. There's differential methylation patterns that clearly separate a low exposure group on the right from the high exposure group on the left. And these methylation data were adjusted for covariates, including age, BMI, sperm motility, time in storage, smoking status, and batch effects. If we analyze the genes that are differentially methylated here, we see that the majority of the differentially methylated genes were found in protein coding regions. Then again, if we look at the differentially methylated genes in both a discovery set of 32 samples and a validation set of 20, and then look for genes that overlap with genes in the Safari autism gene database, we find that by hypergeometric distribution analysis, we find that there is enrichment in ASD genes in both the discovery sample set and the validation sample sets. So the Q values indicate high enrichment in the autism risk genes. If we look at the 14 genes between the discovery and validation sets that overlap with the Safari gene set, we see a number of genes that are implicated in development. So finally, looking at functional and pathway analysis, we found that the DMGs also show significant associations with development of central nervous system, synaptic transmission, hormone metabolism, social behavior, language and movement disorders, autism, and intellectual disabilities. Finally, if we took all of the 52 samples together and look for blocks that are significantly differentially methylated across all of the samples, we find that there is one specific region on chromosome 15 that shows genome-wide significance even after multiple testing correction. And this is the region that encompasses the SNORD 115, which is small nucleolar RNA. This is a non-coding region that's paternally imprinted on chromosome 15. And interestingly, in our studies, DMR's differentially methylated regions involve multiple of these SNORD repetitive elements, and the block above it in green shows that huge block that shows genome-wide significance for differential methylation. Interestingly, the same region, which is represented by the red bars here, was also found to be significantly differentially methylated in sperm from fathers with ASD children relative to fathers of unaffected children in a study by Feinberg et al., published in 2015. So this data collectively suggests the potential for environmentally induced ASD-associated epigenetic alterations that may be transmitted transgenerationally through germline cells. So to summarize here, what we've learned from our integrative genomics path to autism is that by integrating ASD phenotypes with gene expression profiles, we identified a set of genes, including RORA, that can describe the biology behind different phenotypes of autism. Then by integrating epigenetics with the gene expression profiles, we identified this one gene, RORA in particular, that can be regulated by DNA methylation, and further showed that this gene can be regulated by sex hormones as well as EDCs. And finally, we showed by looking at epigenetic analyses of DNA methylation in sperm, we find that pesticides, particularly an EDC called DDE, is associated with DNA methylation changes in sperm, some of which associate with the same sites that are differentially methylated in sperm from fathers of individuals with autism spectrum disorder. Now, at the top of the pyramid is the biggest questions to date. That is, what are the environmental factors that can pose risk for autism? And I suggest that there are probably thousands of them, but we really don't have a handle yet on identifying all of them. What I've described just in this particular talk is just one class of chemicals known as the endocrine disruptors that might pose risk for autism by affecting a gene like RORA as well as other genes that are neurologically that are involved in neurodevelopmental processes. We've also showed that human exposures to EDCs can also be associated with DNA methylation changes in sperm of exposed men that can impact genes involved in neural development and autism. Epigenetic changes that occur in germline cells may be partially responsible for the heritability or transmission of disorder across generations following initial environmental exposures. And thus, it is critically important to understand not only the genetic components of ASD, but also how environmental contributors, including social and psychological stressors, interact with genes, primarily through epigenetic mechanisms to impact health and human development. And then finally, I'd like to acknowledge all the people in my lab, most of them master's students, who have contributed to different aspects of the work that I've described here. And then if anyone is interested in the specific studies that I've talked about, there are selected references relevant to the presented material. So if there are any questions, I'm happy to answer them now.
So this person is asking about sleep disruption. So you talked a bit about this. You talked quite a bit about circadian rhythm and melatonin. A lot of parents, you know, go out and turn to melatonin, and I know that you can't give medical advice. Can you point out some studies that support its use, or have you participated in any research that specifically addressed melatonin?
I haven't, but I know that there are several groups that are. I can't name the groups right off the top of my head, but I know that there are several groups that are studying the effectiveness of melatonin. Then again, because, you know, as I said, autism must be studied in the context of subgroups, it really depends on which subgroup you're giving the melatonin to. I think that if there are individuals that are specifically deficient in melatonin, and those people have sleep problems, then melatonin is more likely to have an effect, not just on the sleep condition, but probably also on behaviors. Because I always say, you know, I'm really cranky if I don't get enough sleep. And think of it, if this is a constant chronic condition, you know, how people would feel.
The next question is about the MTHFR mutation.
There are some people that have those mutations, and they can be helped by, I think, supplements along the folic acid pathway. Right.
Okay. The next question is actually about research funding and barriers. This person has a pretty interesting comment. They said, given the profound findings that you're describing here, it seems like research funding should be available to address these environmental concerns pretty quickly. What do you find as a researcher is the biggest barrier?
Well, research is always a barrier for, funding is always a barrier for research, and it's not easy. I've been fortunate to be funded through the NIEHS, and, you know, I'm grateful for that. But I agree that on the whole, research funding for autism is a lot lower than for other disorders, you know. For example, I think that for autism research, there are relatively limited organizations that one can turn to for funding. Obvious one is NIH. Of course, the Simons Foundation, but there again, the Simons has specific areas that they're seeking proposals on. If you don't happen to fall into that particular interest of theirs, you know, you can't apply for funding there. Then there's the DOD, and again, what they're looking for is very specific. So NIH is the only one where, you know, there can be investigator initiated funding to look at a problem that's of specific interest to a given researcher.
All right, so if a parent is interested in pursuing testing to investigate some of these different genetic disruptors in their loved one, where would they do that, and what in terms of treatment, obviously a lot of this is still very theoretical. So what can they do once they have those results?
Are you asking where can a parent go to see whether or not their child's been exposed to different
right. If there are specific tests available right now that would identify some of the findings that you discussed in today's presentation, or
I don't know of a laboratory that will just run a whole bunch of tests to see what there are just thousands of chemicals out there, and so the question is, what set of chemicals would any given lab be testing? And that's why I kind of took you through sort of the path of discovery of how did we get to the endocrine disruptors in the first place. And because of the sensitivity of one of the genes that we've discovered to be associated with autism, that is RORA, the sensitivity of that gene to the sex hormones, where the endocrine disruptors tend to interfere with the proper functioning of sex hormones in the body, that's what led us to this class of chemicals. But I'm sure there are many other classes of chemicals that could be risk factors for autism. But the question is, how do you identify which ones those are? And there are a number of studies who are groups that are looking at different types of environmental agents, including air pollution. I know that's a biggie, that a number of groups are studying air pollution as risk factors for autism.
Right. So a parent right now can't just walk into a doctor's office and ask for a microarray analysis, right? And
no.
That's not something that we can do right now. But certainly they can sign up for research. So if studies are occurring, they can certainly look into enrolling their loved one in those. So that is one way to gather that information.